real-time PCR. In real-time PCR, the amplification can be checked in real time using fluorescent DNA probes that can bind to DNA amplicons. You should not be confused with RT-PCR, which is also called as reverse transcription PCR or semi-quantitative PCR, because in that one can use only RNA as template, which is converted to cDNA by the reverse transcriptase enzyme. Real-time PCR is also called as qPCR. It is also called as qPCR. Q because it is quantitative PCR. It's going to tell you how much of the DNA or RNA is present in the given sample. You can also compare the levels of gene expression between the given two samples. You can check the presence or absence of any pathogen. You can also check how much of pathogen is present in a given sample. Ultimately, it is the same methodology as in semi-quantitative PCR. In semi-quantitative PCR, the reaction, you see it on the gel and it's a final product where you see the difference between the two samples in the leaf and the root. You see the difference in the expression level of sucrose transporter 1, 2, 3, 4, 5 and actin genes. So now, how is it different? Why we need a real-time PCR when we have a semi-quantitative PCR, which is more simple and more cost-effective? PCR has advantage when we want to know the exact amount of pathogens present in our body. For example, you and I may also be positive for COVID-19, but those who have higher load of virus will show the symptoms of the disease. Generally, there are three phases: exponential phase, linear phase, and plateau phase of gene amplification in a PCR. The exponential phase, the linear phase, and the plateau phase are the three important phases that happens in the PCR machine. Mostly, the template is doubled proportionately in the exponential phase. Means if there's one virus, it becomes 2, 2 becomes 4. So the amplification is in the order of 2 power n. And in the linear phase, what happens is it goes slowly down and uses up all the ingredients present in the PCR tube, and it reaches the plateau phase at about 30 to 35 cycles. So nearly at 40 cycles, everything is used up, and whatever is present is the final end product. That's why in PCR, there is a chance that even if 10 copies present or 50 copies present, both you can see as one single band of the same size. Here you cannot say whether it was initially 10 copies or it was initially 50 copies of the virus present in the blood sample. Because after all the 35 cycles, when you run on the gel, you will still see them as two bands, two blotches. So it is difficult to tell the viral load present in the patient. You can only say the virus is present or not using endpoint PCR. But of course, if the viral load is really huge and you have 10 copy and 50,000 copy, of course, you will see difference in the band size, because that's how semi-quantitative PCR works. Here you see in root, you have a thinner band, and leaf, you have a thicker band. That means in root, the concentration of the mRNA of sucrose transporter 1 is really less as compared to that of leaf. That's why you see some difference. But if you have a minor difference, then you cannot see them on the gel. You may have this question, ma'am, can we stop the PCR machine at the end of the exponential phase here? Can we stop it here in about 15 cycles or so? And can we load on the gel and check? That time you will see some difference in the gel size, band size. Of course, but there are some limitations. First of all, we won't know at which cycle you will have sufficient amplicons that can be detected, because you won't know how much virus is present in the given sample. Only if you know the load, you can tell, okay, at 15th cycle, you may see some band. But if the viral load is really less, you will see some bands only at 25 cycles or 30 cycles. So in such a case where you are really blind about the initial knowledge on the viral load, then you cannot stop at any given cycle and you cannot take them and load on the gel. Secondly, only about 50 nanogram can be detected on the agarose gel. If it is less than 50 nanogram, you won't see any good band or which is fluorescing using ethidium bromide. You cannot see on the agarose gel. So at the end of 35 to 40 cycles, you can see both the 50 copy and the 10 copy as single band size. That's why you have to choose real-time PCR, where you can easily say how much sample is present at the 15th cycle, because in real-time PCR, we can use a dye like ethidium bromide. You have a dye that can mix with double-stranded DNA and it can fluoresce, which can be detected by the machine's fluorometer. And it can tell you, based upon the fluorescence, it can tell you how much of the initial virus is present in the given sample. Not only virus, any DNA can be detected or any RNA can be detected after it is being reverse transcribed into a cDNA. RNA as such cannot be amplified here. Any RNA is very unstable, so we have to convert it into a cDNA and then we have to do a real-time PCR after the RT-PCR is done. So RT-PCR means a reverse transcription is done, after which cDNA is done. And from that cDNA, you can do a real-time PCR. In real-time PCR, there are about 3 types of dyes which can bind to DNA. One is cyber green, then you have Tachman probe, or you have molecular beacons. There are more fluorophore chemistries also, but these 3 are mostly used. And among these 3, cyber green is more cheaper as compared to Tachman probe, and thereby we use more cyber green. And if you want more specificity, we go for Tachman probe. So what is the cyber green? Cyber green means SY stands for Synergy SY and brand. It's from company Synergy, and that's why cyber green. As the name indicates, it fluoresces in green color, and this dye can bind to double-stranded DNA only. As and when your amplification happens, in denaturation process, the DNA gets the double-stranded DNA becomes single-stranded. At that time, the cyber green is moving away from the DNA, and when double-stranded DNA is formed, then the cyber green dye binds to it. So as your amplicon concentration increases, then your cyber green fluorescence also increases. From that, you can say you are getting more fluorescence. That means more amplification is happening. Based upon the fluorescence, you can say how much of amplicon is present or in the given sample. And next thing is Tachman probe. This is more specific and advanced than the cyber green dye. The probe, here it's called as the probe, or the small segment of DNA fragment. This is very complementary to the target sequence. If the target sequence is ATGC and your probe you're preparing as a complementary sequence, this probe is prepared by us. We choose the target sequence, which is very unique and is not present in any other gene of the entire organism's genomic DNA. And we choose using primer blast, and we design this probe and give it to the company, and the company uses this target as probe and attaches 2 molecules. That is Q for quencher and R for reporter. Reporter is nothing but a fluorophore, a fluorescent molecule which can give out fluorescence. And this fluorescence can be quenched easily by another molecule which is very next to it. So that is called as the quencher. So when quencher and reporter are very near proximity, then you won't see any fluorescence signal coming out of those 2 molecules. So then how can we use this as the dye? Here we use Taq polymerase, which has 5' exonuclease activity. 5' exonuclease. Now your primer, one of your primer binds here and as it gets extended, then the 5' end of this probe gets digested by the exonuclease. It chops off the phosphodiester bond in the 5' end and it gets cleaved. The reporter gets cleaved from the quencher because the attached DNA molecules are being chewed off. So it is being fragmented. So your quencher and the reporter are very far apart. When they are far away, the reporter's fluorescence can be easily detected by the fluorometer present in the real-time PCR machine. So this is important method, Tachman probe, and this is more specific than your cybergreen because cybergreen binds to any double-stranded DNA present in the present in the PCR tube. So in the PCR tube, whatever double-stranded DNA molecule is present, your cybergreen will bind to it. The disadvantage of the cybergreen is because even primer dimers, you know what primer dimers are? Primer dimers, when two primers are complementary to each other, then they bind to each other and they form a dimer. Then you call them as primer dimers. And while designing, you have to make sure that your forward and reverse primer should not have these primer dimers. And in case of Tachman probe, since you have a specific probe that is complementary to your target DNA, then such unspecificity can be avoided. So whatever is amplified, whatever signal that comes out is directly from the amplicons. Even if there are primer dimers present, it will not be highlighted here. Next is molecular beacons. So molecular beacons, it is same like Tachman probe, except that the probe is like a stem loop structure. Stem here and the loop here. The loop DNA sequence is very complementary to the target DNA or the amplicons. Here the loop DNA sequence is complementary to the target DNA sequence. So as it binds to the target, the quencher and the fluorophore are apart. Otherwise they were together. The fluorophore and the quencher, they are far apart, and that's how the fluorescence can be detected. But this is more tedious to design the probe, so people stick on to cybergreen method or Tachman probe method. Now, what is the method by which the fluorescence is converted to some value? So just by fluorescence, we cannot tell how much of virus is present. We need some value to convert into the viral load. So we use something called as the threshold cycle or the CT. Here you can see that there's something called baseline. Then you have the threshold. Then the point at which the cycle, number of cycle at which the fluorescence crosses the threshold is called as the CT or the threshold cycle. In the initial PCR cycles, in the exponential phase, the fluorescence signal from the amplicons is not very detectable. So here you have some noise, baseline noise. So that is not detectable by the fluorometer. When it can be detected, as and when your amplification happens, as and when your number of amplicons, number of PCR products gets amplified, only then you can see some fluorescence coming from the fluorophores, and it's not quenched by the quencher. This noise is called as the baseline. This noise is called the baseline. The signal that can be easily detected at a certain PCR cycle is called the threshold value. So there will be a minimum cycle at which the fluorescence can be easily detected by the fluorometer, and that is called as the threshold value. Now this threshold value is constant, and it is constant for all the samples. For in the PCR machine, when you're running a PCR, real-time PCR, this threshold value is set by the machine. And when there will be many samples that you are working with, sample A, B, C, D. So sample A will have a cycle at which it crosses the threshold, and sample B will cross this threshold value at a different cycle number. So threshold value is the point where the signal of the fluorophore is detectable. Now, what is CT is the cycle number. The cycle number. You have different PCR cycles. Each cycle, one cycle means it is having a denaturation, annealing, extension. Once it completes these three steps, then it goes to another cycle of the same steps: denaturation, annealing, and extension. The cycle number at which the signal reaches the threshold value is called the CT. The cycle number at which the signal reaches the threshold value is called as the CT. For example, in this picture, you see two samples. So you have sample A and sample B. Okay, a sample of a patient A, sample of a patient B. Since we are in pandemic, we can always take the example of coronavirus. Here, in the they have set a baseline. The machine has set a baseline, so threshold value. So whenever your signal fluorescence crosses this threshold value, then you call it as CT. In sample A, because the sample is strong positive, the CT occurs at a very early cycle number. Means the more the coronavirus, it is easily detected at the less number of cycles. So at the cycle 21 itself, you are getting so much of amplification that its fluorescence can be detected in the fluorimeter. And in case of sample B, you see the cycle at cycle number 39 only, the detection of fluorescence is happening. That means this person, this sample or the patient B has a very less viral load, but still he has it. That's why you are seeing an amplification at 39 cycle, and the CT for sample B is 39 and CT for sample A is 21. So now we can say what is the viral load based on the CT. Now we know that sample A has high viral load and sample B has less viral load. But from this picture, we cannot say how much exactly the virus is present in sample A and sample B. That's why we go in for absolute quantification. So absolute quantification tells you how much copies of the RNA or DNA of a virus, plant, bacteria is present in samples, and that is useful especially in detection of pathogens. What is the difference between absolute quantification and relative quantification? In absolute quantification, we can tell the quantity, amount of the DNA or RNA present using the standard curve. In relative quantification, you can say how much of the gene has been expressed. Is usually relative quantification is done for checking the expression of any gene in sample A and sample B. And you can tell how much of gene expression or mRNA has been transcribed from gene A in sample A, and how much of mRNA of gene A is present in sample B. And you are using a control sample here. Between the two samples you're checking, and thereby you are using a endogenous control. In endogenous control sample, that means I told you in semi-quantitative PCR, you're using a gene whose expression, the mRNA that is being transcribed from the particular gene, has same level of expression in all the tissues, in all given points, in all given environmental factors. So that basically the gene, if it is producing 10 copies in leaf, it produces same 10 copies in root. So you're nullifying your RNA isolation, your gene X, using this particular endogenous control. So only if your endogenous control has equal concentration in both sample A and sample B. So in leaf and root, if you're isolating RNA, you should make sure there are 10 copies in leaf and 10 copies in root of the endogenous control. Only then we can say that you have taken equal concentration of RNA then you can study the expression level of gene X. So gene X can vary. It can be 5, it can be 1. So it may vary depending upon how much it is being transcribed. So even if you by mistake you're taking a different RNA levels in root and leaf, it gets nullified. You minus 10 minus 5, 10 minus 1. There is a formula to nullify the minor errors in RNA concentration because it gets nullified using your endogenous control genes. So for where we want to use this absolute quantification, for example, correlating viral copy number with the disease state. And here if we want to check the gene expression in response to a drug. So you give a drug, for example, Randomizumab, some drugs that are being given to coronavirus, then you can see what is the expression of a particular gene in a patient sample, in sample A and sample B. So you check the gene expression after giving some drugs or after giving some treatment in response to some factors. How is this absolute quantification done? You must have studied in colorimeter. In colorimeter, what we do, if we want to know the concentration of blood glucose in an unknown sample or a patient, then we make a serial dilution of the substance and we plot a graph. So for example, if you want to check glucose, we take 1 microgram, 2 microgram, 3 microgram, 4 microgram, 5 microgram in different tubes. And we do a colorimeter analysis and we keep the tube, and then the light passes through the colorimeter. Whatever OD value we get, we use it in the standard curve and then we plot it. And if you have a patient sample and you want to find out how much glucose is present in their blood sample, then what we do, we plot that standard graph here and we find out that the person has 3 microgram of blood in his, 3 microgram of glucose in his blood. Same way in real-time PCR also it works. If we know the viral sample, we have the viral sample in hand, what we do, we take the serial dilution of the virus in 5 tubes. One serial dilution in terms of 2, 4, 6, 8, and 10. Then then we plot a standard graph. The real-time PCR machine itself plots the standard graph by checking its fluorescence that comes out of this 2, 4, 6, 8 samples, and then it plots a graph here. And then we also load another sample, which is sample X. We want to know how much virus is present in sample X. Then the real-time, whatever fluorescence that comes out is plotted against the standard graph. And here you can, the machine itself will tell you, you don't have to do anything manually because all these are automated. And at 7th point, you can say that it is having 10 power 7 viral load. This is absolute quantification. Now, there is something called as relative quantification. In relative quantification, we can find out what is the expression level of gene A as compared to gene B in a given tissue. Here, you can say what is the level of expression of sucrose transporter 1 in leaf and root. We can do this in semi-quantitative PCR. As I told you, semi-quantitative PCR is an endpoint PCR. So you cannot use that as the complete foolproof method. So we go in for real-time PCR. When in real-time PCR, the fold, how much of the expression level is changed in root and leaf is given as fold change. The fold change is measured. So since you are checking the level of the gene expression of sucrose transporter 1 in leaf and root, we need to find out by a formula to check how much is the difference, whether it is twofold higher in leaf or threefold higher in leaf or fourfold higher in leaf. How can we check? Here you can say that it's 0.4 and it's 1. So it's almost twofold higher in leaf as compared to root. How can the machine tell you this and plot a graph like this? Is using the formula 2 power minus delta delta CT. So delta means difference. So the difference you have made, you have to make a 2 times difference. One is CT of gene of interest minus CT of internal control, that is endogenous control. Endogenous genes control in sample A. Here the sample A is leaf and the sample B is root. So you take a root tissue and leaf tissue, that is sample A. In leaf, you are checking the sucrose transporter 1 and the endogenous control is actin. So you're subtracting the expression from actin just to normalize what in from the endogenous control to make sure you are taking equal RNA concentration. So it is for normalization. You nullify the effect of the internal control using this formula, and and you subtract this from CT gene of interest. So here also the gene of interest is sucrose transporter 1 in root, whereas that was in leaf. And here also in root also actin will be present. So you're taking the internal control in root as actin gene. So you're subtracting this. So you subtract the entire from the leaf minus the root, that will give you the fold change. So how much of sucrose transporter is present in leaf as compared to root. From the picture that is given to you, I can say that the root leaf has twice the expression level of sucrose transporter 1 as compared to root. This is how you will do your experiments to find out the relative expression of any gene in any given sample. You always have to remember that you have to include the endogenous control to normalize the RNA concentration, just to make sure that you have a same RNA concentration from the beginning in 2 samples. Both the samples should have the same RNA concentration. We already discussed about this fluorophore chemistries, where we saw about Tachman probes, molecular beacons, cybergreen. There is something called melt curve analysis when we want to use cybergreen as your dye, because cybergreen is non-specific. It does not bind to any specific DNA. It binds to any DNA fragment which is double-stranded. So even if there is a primer dimer, it will also be considered as a fluorescence. So how can you detect if there is a primer dimer or not? If you don't have any primer, there is something called melt curve analysis. After the end of 30 cycles, you subject the PCR, the machine itself subjects the PCR tube at 95 degrees Celsius, where the double-stranded DNA melts to single-stranded DNA. Then what happens is, now all the fluorescence will be quenched, because only if it binds to double-stranded DNA, then it will be able to fluoresce. If it is coming as a single strand DNA, then the fluorescence will not be seen. Then again, you allow the annealing to happen. In this case, if you see a 2 fluorescence peak, then that means one major peak is from your amplicon and the smaller peak is from primer dimer. Whereas here you see only one peak, that means there is only one amplicon and there is no primer dimer. So this is important factor when you have to consider while doing your real-time PCR. Tachman probe we already saw, it uses fluorescence and quencher, and it's more specific and mostly used by people when you're studying with 1 or 2 genes. It's very easy to design your probe yourself and do a specific PCR reaction. And molecular beacons is not used as much as those other two. You can do a multiplexing PCR also, where instead of doing one reaction in a tube, you can look for both endogenous control and the target sequence, that is gene X and the endogenous gene like actin and sucrose transporter 1. Both can be checked in the same tube by using different dyes for each target. So quencher 1, reporter 1 for gene X, quencher 2, reporter 2 for endogenous gene actin. So in this way, you can design your probe and you can put it in the same tube and you can check, do a PCR reaction. And now, real-time PCR applications. We were always discussing about quantification of the virus, quantification of the gene expression, and array verification means you have entire set of 40,000, 50,000 genes that can be checked in one go. And drug therapy efficacy, how much drug you're giving and whether the patient is responding to the drug or not. DNA damage measurement can be checked. And then quality control and assay validation. You can be checked in food products. Pathogens can be detected. Genotyping, whether mutation is present or not, can also be checked using real-time PCR applications. This real-time PCR is one very good technique which has come out in the recent times, and when you get chance, you all should go for a workshop in real-time PCR applications, and that will change your entire idea of biotechnology or genetic engineering. And it's a very important technique for you to learn hands-on.